Hello, my name is Adams BT, and a friend or colleague I'm going to do this presentation with is Kalim Ahmed. Today we are going to review an article titled Hepatic Expression of the Human Insulin Gene Reduces Glucose Levels in Vivo in Diabetic Mice Model. So the aim of this article is to evaluate the human insulin gene expression following intraliver plasmid injection in diabetic mice as a possible approach to gene therapy in insulin-dependent diabetes mellitus. Insulin is a hormone secreted by beta cell of the pancreas, which charge to control the production of carbohydrate and fat in the body. The level of insulin in the body is in constant proportion to maintain the right amount of blood sugar in the body. The resultant effect of fall in insulin level in the body is the development of diabetes mellitus. There are two types of diabetes, type 1 and type 2. Type, tw type, type 1 results from the loss of insulin producing beta cells of the pancreas. Type 2 is characterized by insulin resistance and relative insulin deficiency, and it is not non-insulin dependent. Insulin injection is widely used for the treatment of type 1 diabetes. However, gene therapy may provide an alternative hope for the treatment of insulin-dependent diabetes mellitus. Another possibility is intraliver injection of DNA construct, expressing insulin with the aim of increasing insulin levels and ultimately reduction in glucose level. The study was conducted on two different experimental systems. In vitro using 3 to 3 fibroblast cells, in vivo in diabetic mice. In both approaches, DNA constructs were in both approaches, DNA constructs expressing the human insulin gene were overexpressed using plasmid containing promoters and enhancers of cytomegalovirus, that's into bracket CMV, and human hepatitis B virus. The 3 to 3 fibroblast cells were transfected with the insulin gene using the calcium phosphate method. Changes in glucose concentration were observed using glucose oxidation method and cell culture medium analyzed 1, 2, 3, 5, and 7 days after the transfection. In the in vivo studies, the diabetic mice males of 2 months old were used and were generated through streptozotocin method for the generation of diabetic mice. The DNA construct carrying the insulin gene were injected 2 weeks after first streptozotocin treatment. Glucose levels were measured starting from 6 hours after injection and continuing every 24 hours for 7 days. Data were analyzed for statistical significance using two-tailed students t-test for paired data. Differences with p, that's probabilities, values less than 0.05 were considered significant.
My name is Kalim Ahmed and I'm taking over from Adam's BT and I'll be presenting the results and a better expression levels of the insulin gene were observed and a similar experiment as in the CMV promoter was performed. Similar results were obtained as you can see. In conclusion, secreted into the blood flow from hepatocytes transfected with expression vectors and the proinsulin could be responsible for the reduced glucose levels in diabetic mouse. The paper, however, suggested that more studies should be carried out into this establishment, the real facts as to what is happening. In this study, where the expression of insulin was not regulated, there was the issue of hypoglycemia in mice after the expression vector was injected. Hence, to overcome this in future constructs where the expression of insulin is under control of blood glucose levels should be employed. In conclusion, the article shows out clearly that in vivo diabetic gene therapy is possible. Also, the data demonstrates that the reduction in glucose levels in vivo in diabetic mice after treatment with plasmid expressing proinsulin may be useful in gene therapy of diabetes mellitus type 1.
So that is the end of our presentation. So thank you very much.